Well, ladies and gentlemen, let's get started. It is Tuesday, and we're going to be looking at Chapter 19 over the next two times together. And this will be the last chapter that will be part of your next exam. The next exam is on the horizon. It's next Tuesday, and that will be in the blue and gold room as they have been. That'll be a full-length exam. Again, I've already pre-recorded a lecture on blood. I wanted to make sure I had good quality time to discuss the cardiovascular system because much of what we'll be discussing in the heart is brand new. At least it hasn't even been mentioned in 105. The blood story, largely review, plus we've been through it in lab as far as identification of the different leukocytes. So I think you'll find it's a 1-hour lecture. Somewhere in the middle of it, I had to stop and take a break. And when I picked up, I think I repeated about 5 slides. So I apologize, but I realized, oh, I've already been here once already today. And so you might find in the very middle, there's a little bit of a repetition, but it's about an hour of material. And that material will also be on the exam. So you'll have Chapter 11 over skeletal muscle. That last little bit compares cardiac and smooth muscle. And then there'll be the chapter on the blood. And finally, today and Thursday, the chapter over the heart, Chapter 19. I will ask that you look through the first 45 slides on your own. I'll flash through them right now, but I think you'll recognize this is largely a review. If you are not comfortable with the flow of blood through the heart, if you don't recall how blood comes into the right atrium and goes through what valves and gets to what chambers, then you would want to definitely spend a little bit more time on these first few slides. Also, in the first few slides, I have the different vessels, arteries, and veins that you should recognize as well. So let's get started with Chapter 19. Are there any questions just logistically? Remember that your lab reports are due this week in lab for peer review, and that they will be due next week as well. So the Tuesday students are already looking at me saying, Mac, you've got a lab report and an exam on the same day. I realize that. So when you finish your peer reviewing, just get it done quickly, be done with it, and then start focusing on the exam. Thursday students can wait a little bit, right, and get their lab report done after the exam next week. The case studies are also due this week. I'll accept those through Thursday. So hybrid students, I'll accept them in lab Thursday night. The rest of you should have them done by Thursday when I see you in lecture. Remember, too, that one person in your group will be uploading the body of your answers to SafeAssign, just one person per group. Make sure that you include your names at the top of the paper so that when I get that document on the other side, I can line up who was in your group. Other questions, though, related to scheduling or anything that's going on in the lab or lecture? Mary? 11, 18, and 19 for the exam, yes. 11, muscle. 18, blood. 19, heart, yes. Those will be the 3 chapters. Now, wherever I get through on Thursday will be the end of the material. I anticipate getting through this chapter, but if I'm cut short a little bit, then we'll deal with that on Thursday. So again, you should be able to go through the heart and know that there's a pulmonary and a systemic circuit. And if anyone would rather teach this, I'll be quiet and wait for you guys to come up here and teach this. Thank you. So you should know that there's a pulmonary circuit and a systemic circuit, and the pulmonary side of the heart is the right side, the systemic is the left side. And you can watch these little videos. Remember that the left side of the heart is the stronger side. It has the much thicker left ventricle, which is going to push oxygenated blood out to all the organs via the aorta. The right side of the heart is not as strong. It is receiving blood from your systemic circulation. It is primarily only pumping blood a short distance to the lungs. So the right side is your pulmonary side and is largely involved with just getting blood over to the lungs to get it reoxygenated. This shows a nice picture of this, and I don't think I clarified too much in 105, but if you look at the base of the heart, we're looking right at the apex. Okay, right at the apex. And what you'll notice is that the muscle fibers of the heart are actually arranged in a spiral orientation. So that when the left ventricle is contracting and as it's squeezing, it's not just squeezing, if you will, like, I don't know, just squeezing a ball, but it's actually twisting and squeezing simultaneously. So this is pushing blood up and out, right, the pulmonary valve and the aortic valve as it's leaving the left, or leaving the ventricles. So You don't appreciate it from looking at on the side. You can see, I guess, some spirally appearance here, but you can really notice it looking at it from the bottom with the apex right about here. So you know the four chambers of the heart, you know that there are valves, and I'm going to blow right through this. If there's something that you know, hey, I never quite got this before, please let me know. In lab this week, you'll be reviewing really not so much the structures of the heart. You'll be looking at the cardiac conduction system of the heart, looking at the electrical system within the heart. But in doing so, you will have a heart model in front of you. And if you find that you need to go back and review, oh, those are my chambers, those are my valves, please take a few moments to do that. It should come back rather easily, I hope. So there are the atrioventricular valves. They are, as the name suggests, found between the atria and the ventricles, and they are held in place or kept closed through those chordae tendineae, the tendinous valve, the tendinous cords that we know about. Then there are the two semilunar valves, and these are the valves that are controlling the blood leaving the heart, either going to the lungs or to the aorta. If you cut the heart in exactly the right plane, then you would find all four valves in the same plane. Those valves are embedded in a connective tissue layer referred to as the fibrous skeleton. So the fibrous skeleton is both a structural component of the heart, giving structure and helping all the valves to stay in the right orientation. It also creates an insulation, because as we'll see in a moment or today, that when the right and left atria squeeze, when they contract, there has to be a necessary hesitation before the ventricles contract. And part of that is the electrical system needing time to send that signal, but there's also basically an insulation that is provided by the fibrous skeleton. When you're looking at this, you can really see the AV valves, and you can appreciate the chordae tendineae that are attached to it. And then for the aortic valve and the pulmonary valve, those are the two semilunar valves. You can also appreciate, I think, by looking at this, that of the two AV valves, this is the tricuspid. There are three parts to this valve, whereas for the bicuspid, there are really only two flaps. And that's where they get their name. Remember, cusp means point, so there's 2 points or 3 points in that valve. Next week, when we have lab, we'll be doing a heart dissection. Now, in 105, you did a heart dissection. We did a quick one when we were doing the plucks. But now you'll probably look at the heart in a new way after we go through this discussion. So I want you, freshly dissecting through a nice heart next week. And as you do so, think not only about the structures that you already know about, but some of the new ideas that we'll be adding to your understanding today. Beautiful view. You can see how floppy, if you will, those valves are. They're not as, quote, structural as you might imagine a valve to be. So when you're doing your dissections, you may have a difficult time, depending upon the slice, of seeing the valves. But I think you'll appreciate that when you're doing that. Here you can see light behind it, really showing, here's a papillary muscle, and here are the chordae tendineae, the tendinous cords coming down, and you can see the opening of the valve as well. Now, you should know the flow of blood through the heart, the order in which blood goes through the heart, and what we need to appreciate is that blood only moves from one chamber to the next when there's a pressure change. These valves are not opening and closing like automatic doors. The valves are being pushed open, and they're only being pushed when there's a pressure change. And what's causing that pressure change is going to be the squeezing of the heart muscle. So when the atria squeeze, what's being pushed open? When the two atria squeeze, what valves are being pushed open? The AV valves, right? The AV valves going down to the ventricles. And then when the ventricles squeeze, and they do that spiral squeeze, they squeeze and rotate, now what valves are being pushed open? The semilunar valves, right? The aortic and the pulmonary, or you'll hear pulmonic or pulmonary valve. So remember that those valves are not just opening and closing on a timer, right? That they are being pushed open by pressure changes as blood is being forced through those different valves. So the ventricles will relax, the ventricles will contract, and we're going to have, again, an alternating system of relaxation and contraction. The contraction will be called the systole or the systolic, right? The time of contraction, the squeezing pressure, and the diastole is the relaxation or the diastolic pressure. And we'll discuss the systole and diastolic pressures as we move forward as well. So the 11 steps basically that you need to be thinking about are here. So I would definitely recommend, if this is not something that you can recall easily, go back and take a look in chapter 19, find this image in Saladin, and review those steps as the blood is coming in and flowing through your heart. Very important. As you're doing it as well, think about, is the blood blue or is the blood red? That is it freshly oxygenated or is it instead less than fully oxygenated blue blood? Any question about that? Any thought or concern or anything that you remember, man, I never quite got that, or I need to have something clarified when it comes to just flow of blood through the heart, the names of the chambers, the names of the valves? Remember, if you're going from the right side, the valves, you're going to try it before you buy it. So the tricuspid is on the right side and the bicuspid is over on the left side. One thing that we didn't discuss very much last semester, though, is that, of course, the heart is pumping blood to the rest of your body, but the heart is also pumping blood to itself, and that there are coronary arteries that are going to be branches off from the aorta, and those coronary arteries are then going to bring fresh oxygenated blood to the heart muscle itself. So about 5% of all the blood that is pumped from the heart is immediately redirected back to the heart muscle. The blood going through the heart is not providing oxygen to the heart muscle, right? It has to come out the aorta, as you'll see in a moment, and then circulate around in the coronary arteries, and then that blood is bringing oxygen to the heart muscle. This means that about 250 mils of blood per minute. We'll look at some numbers as we go through this, but 5% of your blood volume, or about 250 milliliters of blood every minute, is going back to your heart. And your heart is very, very finicky. It needs freshly oxygenated blood. It does not survive at all well without oxygen. The heart cannot run in anaerobic states. So you know a little bit about aerobic and anaerobic. The heart must have oxygen. There is no capacity for it to operate in an anaerobic state. So it must have a constant flow of oxygen and glucose, because we need to be making ATP, right? And contraction of the heart is very costly from an ATP standpoint. A couple structures here that are new for you, and I'll just ask you to go back and look at them. But in 105, I just described the arteries as the coronary arteries, right? We just named them the left and the right coronary arteries. Well, those left and right coronary arteries split into other smaller arteries, which are, of course, it's anatomy, they're named. So the left coronary artery, the LCA, branches off, and you'll see it branching off the ascending aorta in a moment. And it's going to branch into the widowmaker, which is the anterior interventricular branch. Okay? And this is going to bring blood to both ventricles. So, why is it called the widowmaker? Because when this one goes wrong, you're losing blood supply to some very critical portions of the heart. So if the widowmaker gets blocked and you're not getting blood to both ventricles and to a lot of the interventricular septum, what's going, for those who had lecture or lab today, what's going down the interventricular septum? The bundle branches, right? You'll see that in lab this week. So there's a lot going on there. So if you're not getting oxygen to your ventricles, this is not good, right? It's not likely going to be a good outcome to the heart attack, so we get the name widowmaker. There's also the branching of the L of the left over to the circumflex branch, and you'll see that this then goes off to the left marginal branch and ends up on the back side, on the posterior side of the heart. Pictures in a moment. So you can see here, these are from Anatomy and Physiology Revealed. I would not have you label these, so I don't want you to stress out about labeling images here, but do know about these different vessels. Okay, and then you can see that it ends up on the posterior side, the left side does. On the right side, the right coronary artery branches off and goes to the right marginal branch and to the posterior interventricular branch. And so clearly, if you have a problem on the right side, clearly you've got a heart attack, right? You've had a blockage of blood flow to the heart, and you're going to have damage to the heart, but you may more likely survive this heart attack because it's not affecting the ventricles as directly. Also, this right coronary artery goes to the pacemaker. So you'll recognize that term when you get through lab this week. But the pacemaker is a portion of the right atrium that sets the heartbeat. And the good news is nowadays, if you had a heart attack and you damaged this portion of your heart, what do? We give you an artificial pacemaker. So it's possible for someone to have damage to this part of the heart, survive that heart attack, and then have an external pacemaker put in to keep the heart beating regularly. So again, you can just look at where these arteries are branching off. I think it's just as easy to see it, probably easier to see it on this image. So do look at these cartoon images when it comes to labeling or naming these arteries. So we're coming off the aortic arch. Do you agree? Right, we're coming up, aortic arch. And as soon as we come off, you can see right here on the right side, this is going to be the right coronary artery, and it's going to split as described. And the left side we can't really see, but it's going to come around, coming down the branch here, coming down. Do you agree that this would be this is the apex? So this is what ventricle? That's the left ventricle, isn't it? This would be the right ventricle. We kind of lose perspective here, but back underneath all this, that would be the right atrium, and back over here would be the right, or sorry, the left atrium. Looking at it from the posterior view, remind me, what is this structure? Coronary sinus, and what's the significance of the coronary sinus? All the blood coming from the heart muscle, right? It got there from all these arteries, is going to drain back through a series of cardiac veins, and all those cardiac veins drain into the coronary sulcus, and the coronary sulcus has its own direct route back into the right atrium. So this blue blood coming back from the heart can directly go into the right atrium, which is not there, but okay. Over on this side. So what is an MI, a myocardial infarction? An infarction is a lack of blood supply going to the heart. So it's a branching, or it's a blockage of any of these coronary arteries. If it's a large blockage, if it's very proximal and it takes away a lot of blood flow to the heart, that's not going to be a very good outcome because you're going to destroy a large part of the heart. If it's in a smaller branch, then the damage would be less and therefore more likely to survive that heart attack. It's going to be caused from a blood clot, right, that suddenly is formed and then goes to the heart, or it could be a breaking off of the fatty deposits that are common in atherosclerosis. Next week in lab, you'll see under the microscope an artery with atherosclerosis, and you can appreciate that buildup of fatty materials, how it reduces the flow of blood through the vessel, and how that fatty buildup can break off and travel and act as a clotting factor. Now, one of the good things about our heart is that there are a lot of anastomoses. And you may recall an anastomosis is a place where there's an alternate route. There's more than one way to get blood to a portion of the heart. If you look at an angiogram, you look at somebody who's looking at their heart under image, and they're pumping some dye in there so you can see all the vessels. You would note that there's a lot of vasculature going around the heart muscle, and that no two hearts have exactly the same exact series of vessels. So there's a lot of overlapping redundant blood vessels, and this is good, right? So that if you have a small minor blockage, there may be another way to get oxygen and blood to that particular area of the heart. So because of the arterial anastomoses, because there are alternative routes of blood flow, we can call this collateral circulation, a way of getting blood around an area to another area. Now, the heart is taking turns, actually. Look at this statement here. Blood flow to the heart, muscle itself, is actually slowed during ventricular contraction. So what that tells us is what? When the heart is pumping blood, and it's pumping it with great force to the rest of the body, that at that moment, there's actually less blood flow going to the heart. So there's this, again, sort of a pulsatile blood flow going to the body that takes turns with the pulsatile flow to the heart. So why is this pulsatory? Well, as the myocardium is contracting, right? As the heart is doing the ventricular contraction, it's actually going to compress down on the coronary arteries. You'll see a picture of this in a moment. But as the heart is squeezing, it's actually going to squeeze down and block flow through the coronary arteries. This is going to obstruct blood flow. Also, when the aortic valve opens up, it actually covers a little bit of the beginning of the coronary arteries, because the coronary arteries come off from the aortic arch just as soon as you get past the aortic valve. So when the aortic valve is open, it's actually blocking blood going over into those coronary arteries. And then when the ventricles are in relaxation, when they're in diastole, then the blood can then surge back. And so basically, as blood is coming out of the aortic valve, at that moment, there's actually less blood going to the heart muscle. And then once the ventricles relax, then that blood flow can kind of come back and go more fully into the coronary artery. There's another condition I want you to appreciate, that is angina or angina. You'll hear it pronounced both ways. This is chest pain that is exacerbated by a partial block, not a complete block. So a complete block is an MI, a very crushing pain usually from that complete lack of oxygen. But if there's any ischemia, any lack of oxygen, not complete, but partial, then there will be a little bit of shifting to the anaerobic fermentation. Now, the heart does not like this at all, and it produces lactic acid, and just like you're having muscle cramping or muscle pain from a building up of lactic acid, the heart sends off a signal saying, hey, I'm hurting, right? So you get this uncomfortable sensation caused from this lactic acid buildup. But the heart can't maintain this very long at all, and is oftentimes a sign, right, of a more significant problem coming. So angina is a partial blockage. An MI is a complete blockage leading to a sudden, rapid death to a patch of the myocardium. Again, it could be from a blockage, from a blood clot, or a fatty deposit, and everything downstream of that blockage will die. And it depends on which portion of the heart and how much of the heart is affected as to the outcome of that heart attack. Will the patient survive it or not? Now, some heart attacks may be, quote, painless. They're not so much attacking the muscle, but they might disrupt the electrical conduction pathway. All right, so Vince, you were asking about that today. What happens, sometimes we get these irregular or these arrhythmias that you'll see in lab, and you'll get into an atrial flutter or to a ventricular issue. And the person they're really not experiencing any pain, but their heart is no longer sending the electrical signals through it properly. The heart is no longer beating like it should, and the heart's basically just quivering. It's no longer contracting. Well, the blood's not moving, right? A quivering heart is not moving blood as it needs to, so the brain's suffering, the rest of the body is suffering, the heart's not getting its oxygen, and eventually, right, just like anything else, if there isn't oxygen going to the heart, then the person's going to have some serious issues. And MIs are a major cause of death, obviously, in our country and around the world. Then we have blood going to the heart. We also have blood coming back from the heart, so venous drainage. So there's a lot of blood coming back directly into the heart chambers, and the rest of it, and that's coming from the Thebesian veins, and then there's also the great cardiac veins. I've never understood why we call it the coronary arteries, but we call it the cardiac veins, but that's just the way it is. Why aren't they both just coronary, or why aren't they both just cardiac? I think it's just to keep us on our toes. So the coronary arteries and the cardiac veins. And all the veins, as I've already mentioned to you, are coming back from the heart muscle and are emptying into that coronary sinus, right? That big blue space that we find on the underside, on the posterior view of the heart. So this is coming back through the middle cardiac vein and the left marginal vein, and again, all comes back up into the coronary sinus, which then goes back into the right atrium. You can see that here, big structure on the bottom side of the heart, that's the coronary sinus, and blood has drained back into it from numerous cardiac veins. Well, that brings me through pretty much the section that I wanted you to review on your own. So please, if that's giving you any trouble at all, go back and spend some more time on the overall pathway of blood. The next few slides are going to be sort of a repeat of what you'll hear in lab this week, which has to do with the cardiac conduction system. How does the heart beat? What sets the heartbeat? And how does that whole thing work within the heart? So we'll start off thinking about cardiac muscle cells. The last thing you need to do back in chapter 11 was read those 3 or 4 pages in Saladin and go back and make sure you can compare and contrast cardiac smooth and skeletal muscle. And the cardiac muscle cells are oftentimes just referred to as cardiocytes, cells of cardiac muscle. And you know that this is striated muscle. You know, however, that the cells are shorter, that they're a little bit thicker, and they oftentimes have wide branches in their arrangement. Typically one nucleus, right? So remember, skeletal muscle is multinucleated. So these are usually, occasionally you'll see a double nucleus, but typically single nucleus in the cardiocytes. And quite a bit of glycogen, which again is an energy storage molecule so that these heart cells always have a good fuel source. You'll always, of course, have the intercalated discs, and the intercalated discs are going to join these cells end to end and create an interlocking. This is going to increase their surface area. You know, it depends on what book you look at and what microscope preparation you look at. Sometimes intercalated discs look like straight lines, right? You've probably seen the straight line kind of intercalated disc. But then, right, you'll see straight lines, but then other intercalated discs look more like somebody drew it with an Etch-a-Sketch. And then other times you'll see an author who will draw it more as looks like the papillary, you know, the dermis and the epidermis coming together. Regardless, this is really not as accurate. At the microscopic level, you have basically an interfolding of one cardiac muscle cell, right? One cardiac muscle cell to another cardiac muscle cell. And this interdigitation is really the intercalated disc, and it creates an increase in the surface area between the two cells. And this is going to allow, again, an interlocking, but also an opportunity for the electrical signals to travel very, very quickly. Intercalated discs are really a combination of two things. They are desmosomes, and you may recall from epithelial discussion in skin that desmosomes were an intercellular junction, basically like 2 parts of a button, right? 2 parts of a snap or a rivet. And each cell gives half of this button, if you will. And these buttons are found in places where there's a lot of stress. Well, would you agree that the heart is under a lot of stress? It's stretching constantly, under a lot of force. So these intercalated discs are a combination of desmosome, right? Something that snaps places of high stress together, along with this idea of a gap junction. That is where something can go across the cells, and that is what when you think about the gap junction, you think about molecules or something being shared between the cells. And so what you have now is an electrical synapse. Remember, here in the cardiac muscle, you don't have a chemical synapse, right? A chemical synapse is one where there are neurotransmitters released, right? And then the neurotransmitters are picked up on the postsynaptic cell and cause depolarization on the next cell. Instead, in heart, you've got electrical synapses. That is, the electrical signal is picked up directly as an electrical signal in the next cell. There's no chemical intermediate. There's no chemical neurotransmitter released. So this allows for the electrical signal to travel much, much faster, right, between the cells than it could if it had to depend upon chemical synapses. Also, if there is damage to cardiac muscle, it does not repair very well. There are really no good stem cells waiting around to repair the damage, and instead, the damaged heart muscle is replaced by scarring or fibrotic tissue. And this fibrotic tissue is not good at contraction, right? So no longer is the heart able to squeeze as hard and push blood out. And so the heart becomes weaker, doesn't it? So a person after a heart attack has a weakened heart. They don't have the capacity to do as much work because their heart will never respond the way that it used to. Again, you'll recognize these images from APR. And at this magnification, the intercalated discs are these little lines that you see scattered throughout the image. And then they do a nice job of sort of highlighting them in blue for you down here. Again, that cardiac muscle, here it looks like an Etch-a-Sketch, right? So there's the Etch-a-Sketch intercalated disc that we see here. And you can appreciate that at the actual intercalated disc, what you have is that interdigitation, right? This waviness, increasing the surface area. You have these openings for the electrical signal to go straight through. And in combination with this, it's like a desmosome in that you've got these buttons snapping the cells together. So I told you that the heart is almost, I think I said never, but almost exclusively aerobic. It does not do well in an anaerobic situation. In angina, I told you that yes, because there's a temporary partial lack of oxygen, some of the heart does flip over to anaerobic, create some lactic acid, and you give a sense of pain with that. But for the most part, the heart needs to stay constantly able to make large amounts of ATP. The way the heart muscle cells can do this is that heart muscle cells are very rich in myoglobin. Remember, myoglobin is a hemoglobin-like molecule found in muscle. It's going to carry the oxygen and keep high oxygen levels in the heart cells. And there's always lots of glycogen around for energy sources, and huge mitochondria. I mean, they're large in size. They're just huge. They take up a huge percentage of the inside of a muscle cell. Now, there is some adaptation. The heart can use glucose. The heart can use fatty acids. The heart can use all sorts of organic fuels, but it must have what? It must have oxygen. So it's not as finicky as the brain. The brain needs glucose and really nothing else to run it. The heart can handle other energy sources, but it still is very, very vulnerable to oxygen deficiency. The heart is also fatigue resistant. Good idea, right? That the heart does not give up on us. Again, it's not fatigue resistant because it really doesn't go into anaerobic fermentation and never has to deal with oxygen debt mechanisms. So we were talking about how in skeletal muscle, right, you're getting an oxygen debt. You start off and the skeletal muscle is not getting the oxygen that it needs. It flips over to an anaerobic state, and then your respiration catches up and you stay within an aerobic state until you're done, and then your body recovers by huffing and puffing and replacing that oxygen for a while thereafter. The heart never has to go through that because it never gets to that anaerobic state, right? It can't. It just can't handle that. Someone asked a question the other day about during exercise. And I told you that in the initial burning of your skeletal muscles, that glucose is all used up, right? Remember that conversation? The glucose is used up very, very quickly. And then we go to the phosphagen system where we make more ATP by other means. Then we get to ATP anaerobic synthesis. And the question came up, what happens to the brain when all the glucose is gone? Okay, and I don't think I answered that question well. It came to me later. The blood glucose is not all gone, right? You haven't depleted all of the glucose in all of the blood. All you've done is depleted the glucose in the immediate area of the skeletal muscle. So see the difference? So when you're exercising hard and when I'm saying you're using up all the available glucose immediately, then going to fatty acids, then going to phosphagen, that's in the localized environment of the muscle. You have not depleted all of the glucose of the entire body, right? Otherwise your brain would be in trouble. Okay, I think I kind of misspoke, or I spoke around that question, and I didn't do a very good job of that last time, so I apologize. Questions so far? So far the story makes sense? So the conduction system that you'll study more in lab this week is what's going to coordinate the heartbeat. It's going to coordinate the rhythm of the heart, and the heart has its own pacemaker. We hear that word used. You know, it used to be that pacemakers were sensitive to microwaves, and so every 7-Eleven would have a sign on it that said, caution, you know, we have a microwave in use, and patients with old-fashioned pacemakers couldn't be around that because it would interfere with their pacemaker. I think we've got to a point now where that's not an issue anymore. But we have our own internal pacemaker, which initiates your heart rate. And basically it's a bunch of nerves, right, that travel through your heart and send this signal from the atria down to the ventricles. Just like when you hooked up electrodes to your arm a couple of weeks ago and you did the muscle grip and you did the EMG and you saw millivolt spikes on the screen, you were looking at electrical activity through the muscle cells. So too, when the heart is beating, there are electrical signals being sent that we can now pick up with electrodes placed on the outside of your body. So we can do an EKG to pick up these signals. And you'll be learning about EKGs this week in lab. Now, the initial signal that comes from the true pacemaker of the heart is called the sinoatrial node, or the SA node. These are modified heart cells. They're not involved with contraction. Their job is to send off this electrical signal. You cannot recognize them differently under the microscope. So there's this little patch of cells. You cannot look at the heart muscle and say, oh, there it is. But there is always going to be a little patch of cells in a normal heart which are sending this signal. And this is going to initiate each heartbeat and help determine your overall heart rate. It's found in the right atrium, right where the superior vena cava dumps into the right atrium. Right about there is where you will find the SA node. That signal is going to spread from the SA node to all of the cardiocytes within the right atrium. It's also going to quickly spread over the interatrial septum and go to all of the cells found in the left atrium. So both atria are going to quickly receive this electrical signal. I'm not getting the sound today, but I can walk through it. So there's the SA node firing, and then the SA node is going to go to a second place called the AV node that I'll introduce in a moment. That AV node signal is now going to hesitate for just a little bit, and then that signal is going to travel down these bundle branches, as you see it going down. It's called the atrioventricular bundle, or the bundle branches, going down the septum, and we'll see it go down toward the apex, and then it will come up around on what's called the Purkinje fibers. Okay, so it comes around, and as this signal is going down, what's happening to the cells? The cells are becoming depolarized, same old story, and those cells, what does muscle do when it's depolarized? When muscle is depolarized, muscle contracts, right? So we'll see that continuation. And it just keeps on going, keeps on going, keeps on going. This, in a normal heart, is happening every 0.8 seconds. So on average, the average person's heart rate is initiated every 0.8 seconds. About 75 times a minute on average. So once the SA node fires, as you just saw in the picture and as you'll label again in lab, it goes to the AV node, the atrioventricular node. It's in between the right atrium and basically the right ventricle. It's down toward the bottom of the right atrium. And it's right there along that fibrous skeleton. So I've already introduced the fibrous skeleton, that connective tissue layer that all the valves are sitting in. So the AV node is kind of right there as well. The AV node, I was thinking, is sort of like being a transmitting station. So the SA node fires, the signal reaches through the atria and eventually activates the AV node. The AV node then picks up that signal and then sends it down what's called the bundle, or named after a guy, the bundle of His. I don't think I use that word today in lab, but the bundle of His, another name for the atrioventricular bundle or the bundle branches, and then to the Purkinje fibers, which go all the way down around the apex and around the ventricle wall, but also go up into the papillary muscles. So that's really all I'm going to say about the cardiac conduction system, since we'll spend a few more minutes with it in lab. I've already said this, I believe, so this is a repeat. We have the cardiocytes. Yeah, I've already got this slide exactly, don't I? So the structure of cardiac muscle cells. Already been here. Interesting. Those two are just misplaced. I do want to show you the picture, though, of the conduction system. And here are the 5 steps to be keeping a track of. So the SA node, number 1, sitting up here toward the superior portion of the right atrium, it fires. Fires usually about every 0.8 seconds. It takes a while for that signal, not very fast, not very long, for it to travel through all of the right atrium. It also shoots across and goes to the the left atrium. During that time, the atrial cells will be depolarized and will be contracting. The signal will reach the AV node, shown here as number 3, and there's always a little bit of a hesitation as it transfers that signal, and it goes through what's first called the AV bundle, or the bundle of His, and then the bundle splits into the bundle branches, and then it continues down around the Purkinje fibers, and that goes up along the ventricular wall, but also goes up into each papillary muscle. So you can see the yellow nerves wrapped around those papillary muscles as well. So that's the conduction system. You'll need to be able to label those components, well, for the test next Tuesday, but also in lab next week. So as you're studying for the exam this week, you're actually over-studying, if you will, for a portion of the lab quiz next week. So next week's lab quiz should be really strong, since you will be way over-prepared studying for this exam. So if you take the heart out, you just take it out of the chest, right? It'll keep on beating as long as there's energy, and as long as you put it into a bath of calcium, right? You better have some calcium available and have some energy available, but the heart will keep beating for a while because of this pacemaker. It doesn't need direct connection to your central nervous system. But the central nervous system does influence your heart rate. It doesn't initiate it, right? The pacemaker, the SA node, is what is telling the heart to start each time. But the sympathetic and the parasympathetic nervous systems are certainly influencing your heart rate. And you know this. You know that when the bear walks in the room, your heart rate goes up. Why? Because in a sympathetic moment, your heart rate goes up. During a rest and digest period, your heart rate slows down. It doesn't slow down to 0, right? Good thing. But it definitely slows down. So we know that there's a rheostat, a control mechanism over the heart, not initiating it, but influencing it. So sympathetic nerves that are coming from, remember, where do your sympathetic nerves originate from? Sympathetic nerves originate from the thoracic, right? So there's some nerves coming from the thoracic segment, and they go through those sympathetic chain ganglia that we know a little bit about, and then they also go to the cardiac plexus. What is a plexus? It's a cluster of nerves that travel together. So you're not surprised that there's a cluster of nerves that are going to go, right, to your heart. Isn't that beautiful? Right, the cardiac plexus. Now, the cardiac plexus, some of those nerves will continue onto the heart and will innervate at the SA node. That's not to say that those signals are doing what? They're not initiating the heart rate, but they are influencing it. So as these signals come through the cardiac plexus and they interact at the SA node, when the bear walks in the room, it's going to cause your SA node to fire more quickly. When you're more relaxed, your heart rate's going to release. It also goes down to the AV node, as I'll describe in a few moments. The AV node also can initiate its own heart rate. Some of it will also go down to the other sections of the heart. Okay. Now, when you are in a sympathetic overdrive, when the bear walks in the room, not only is your heart rate going up, but what else is happening? Your heart's beating stronger, right? I mean, not only is it beating faster, it's like, oh, my heart's jumping out of my chest. Yeah, it is true that not only is your heart rate up, but it's actually squeezing harder. And you experience that during exercise as well. So sympathetic increases rate and contractile force, or the amount of strength that the heart is able to do. And what this also does, though, is when you are, think about it, if your heart rate's going up and you've got more blood coming out, that means your aortic valves are open more often, aren't they? What's happening now to the flow of blood to your heart through those coronary arteries? Remember, I told you when the heart's actually squeezing, the heart itself is not getting blood, right? There's a restriction of blood going to the coronary artery. So when do people usually initiate a heart attack, or when do they initiate an angina attack? Right? Someone's out shoveling snow, right? And it triggers their angina because as their heart is beating faster during that ventricular contraction, there's less blood actually going over to the heart at that moment, and so they sense a lack of oxygen, therefore an angina attack, or a sense of a heart attack, right? Now, not only is there sympathetic going to the heart, there's also parasympathetic. These signals are, for the most part, going to slow the heart, and you know the nerve that travels down to the heart that's part of this parasympathetic signal, and that is the vagus. Okay? Now remember, what cranial nerves are involved in parasympathetic signals? Those are the four nerves, right? 3, 7, 9, and 10. Those are the four cranial nerves that are involved with parasympathetic signals. We saw that before. Just remind me, think about parasympathetic, rest and digest. What's 3 doing? Oculomotor, constriction of the pupil, right? You're relaxed, your pupils are more constricted. 7 and 9, rest and digest, you're preparing for a meal. That's your taste and your salivation. 10, vagus, right, going down to the heart, telling it to kind of relax a little bit. So you've seen those 4 nerves before. So the right vagus nerve goes to the SA node. The left vagus nerve goes to the AV node. It's interesting. The heart is asymmetric, and so too certain nerves. So your right vagus, remember these are pairs, right? There's pairs of cranial nerves. So your right vagus nerve goes down to influence the SA node. Your left vagus nerve goes down to influence your AV node. So let's talk about the cardiac cycle. Cardiac cycle. That is this thing that's going to happen, 60, 70, 80 times a minute throughout your life. And your heart is having to go through this cycle. And there's 2 parts to this, the systole and the diastole, or the systolic and the diastolic pressure. Now, yes, the atria will squeeze, and we'll call it an atrial systole. And the atria will relax, and we'll call it an atrial diastole. That's true. But when you're talking about the heart in general terms, and someone says the heart is in systole, they're usually referring to the greater, stronger vessel or chambers, that is the ventricles. So we'll break it down a little bit, and we will talk about atrial systole, right, when the atria are squeezing, and we will talk about when the atria are relaxed, okay? But typically, if someone's just saying your systolic pressure or your diastolic blood pressure, they're referring to exclusively your ventricular contractions and relaxations. More on that as we get to blood pressure. So a normal heart rate is said to be a sinus rhythm. Okay, they're back in sinus. We've got a nice sinus rhythm. That is the normal heart rate, heartbeat that you'll recognize in lab this week. That normal heart rate will be triggered by the SA node. So your heart, if it's all is well, is firing between 60 and 100 times or so a minute, and this pace is being set by your SA node. That's 60 to 100 times a minute, but the average resting adult's heart rate is between 70 and 80, with 75 being the middle. You'll learn that in the lab. I said 75 is the average. So what I'm saying to you is that the SA node is firing more than your heart rate actually is. And what's slowing down the heart rate? Vagal tone. A fancy way of saying your vagus nerve, and your vagus nerve is part of your parasympathetic nervous system. So you may also hear this called as a vagal tone or parasympathetic tone. What this means is that if I took your heart out right now out of your body and set it in a cold beaker of calcium and it was still beating, it would beat closer to 100 times a minute. It would sit there and beat about 100 times a minute. If I put it back in your body, because now it's being influenced by your overall nervous system, and the vagus nerve is coming down and influencing, not initiating, but influencing your heart rate is reduced down to the normal of about 60 to 70, or sorry, 70 to 80. Now it is possible that other portions of your heart are firing inappropriately. What does the word ectopic mean to you? An ectopic pregnancy where the embryo is out of place, right? It's out of its normal place, where it should be, where it can be. Remember I told you that the SA node, you can't see it under the microscope. You can't look at it. There's no neon lights coming off from it. And it is possible that in a defective heart, that there are other patches of pacemaker-like cells that would be throwing out signals inappropriately. And so that's possible. And this could be from a anatomical problem, or there could be what's called an ectopic focus. And this would be, if you're not getting enough oxygen, if you've got an electrolyte imbalance, if you're drinking too much caffeine, nicotine, other drugs, it can actually cause another part of the heart to fire before the SA node. So you'd have an abnormal rhythm. So, if you drink some caffeine and you hook yourself up to an EKG, you may see some PVCs, some premature ventricular contractions, and that's not uncommon at all. In lab this week, you'll be looking at about 7 different abnormal EKGs. One of them is PVCs, premature ventricular contractions, and one of the things that causes PVCs, right, an extra ventricular contraction, is caffeine or stress. So those can lead to an abnormal heart rate. There's also, and this is where I was kind of leading up to, there are parts of the heart not on the SA node that can cause the heart to contract. These are ectopic foci, a focus, a place. When you see the word foci, I always think about the eye. Light is being focused onto a particular part of the eye. So think about a focus as being a little area, right? An ectopic foci, a little area of the heart that is firing incorrectly. Now, if the SA node gets damaged, the heart rate will be around 40 to 50 beats per minute. Now, is that enough to sustain life? Yeah. Yeah, you'll be okay. It's not ideal long term, but in the short while, you'd be okay. So if the SA node stops firing, right, it normally fires about 75 times a minute. If it gets damaged, then the AV node itself will fire, but it fires more slowly, only between 40 and 50 times a minute. So if you take the SA node out of the picture, the AV node will continue to fire. Do we normally see that 40 to 50? Why don't we see the 40 to 50 of the AV node? Because even before the AV node can fire, the SA node has fired and has activated it, hasn't it? So the SA node is setting the pace. It is the pacemaker. So the pacemaker is firing, the SA node is firing 70, 80 times a minute. If, for example, though, the SA node is defective, it is not firing, the AV node will fire. Now, what if both the SA node and the AV node are not functioning? The heart will beat now at 20 to 40 beats per minute. That's not going to be good, right? Now, you might be long enough to get to the hospital if you were to wipe out both parts, but a person at that rate is definitely going to need a pacemaker put in, right? And probably nowadays, even if just their SA node was firing, if their SA node was broken and they were only firing out their AV node at 40 to 50, they would also put in a pacemaker, just to make sure that there's plenty of heart rate. An arrhythmia would be any abnormal rhythm. So A, right, a negating term. So a lack of rhythm or a lack of normal rhythm would be an arrhythmia. And again, we'll look at 4 or 5 different arrhythmias. This could be from a problem in the conduction system. That is, the heart, there's a block. The heart signal is not able to travel from one portion to another. So the time it takes for the electrical signal to travel either is slowed down or completely blocked. And that's not going to be good. So there can be a partial blockage or even a total heart blockage. That could lead to some arrhythmias. There also can be atrial flutter. And you'll see a EKG of this in lab today. And this is where basically the atria are fluttering, right? They're beating 200 to 400 times a minute. They're just sort of sitting there shivering, right? The atria are basically in this flutter reaction, and blood is not moving very well at all. So what do? We clear, right? Take an AED, shock them back, hopefully get that electrical conduction system back to a normal rhythm. So something's going on in the electrical system of the heart. It's gotten out of whack. We clear them, we overstimulate, and hopefully reinitiate the normal rhythm of the SA node back to a sinus rhythm. I've already mentioned PVCs. You'll see PVC EKG upstairs. And again, those are very common in individuals who don't get enough sleep, stress, caffeine, other stimulants. Every time I go to the doctor, I throw a few PVCs and they say, okay, it's not that abnormal, not to be too concerned, and it's usually right after I had a Diet Coke or didn't sleep enough. So, a little stress, a little caffeine, a little lack of sleep, we'll throw some PVCs out. Ventricular fibrillation. If I'm going to shock you, what am I trying to do? What's the name of what do we call the unit we go get? A defibrillator, right? Yeah. So we're trying to stop fibrillation. So if you have ventricular fibrillation, this is the most serious of all. You've got some serious arrhythmia going on. The electrical signals are not working well. Your heart is not pumping, right? If you have ventricular fibrillation, your ventricles are quivering. They're not contracting. Blood is not moving. Not only is your heart not getting oxygen, but neither is any of the rest of your body. So this is deadly. So if you can't stop that ventricular fib very, very quickly by defibbing, right, adding a strong electrical charge and shocking the system back into a normal sinus rhythm, then death is on the doorstep. Any questions about those? I'm not showing you the pictures here of those. I want you to know about them here, and in lab this week you'll all see those images. Part of your lab quiz next week will be recognizing those abnormal arrhythmias. Okay. Any thoughts so far? How are we doing? Okay. Now, how does this work? Okay, we know there's an SA node. We know that that's what fires, and that signal spreads across the atria and then eventually down to the ventricles and causes depolarization of the cardiocytes. What do we know about electrical signals. For the signal to be sent, there must be a what? A depolarization. And when we were discussing neurons, we said that there was a resting membrane potential of -70, stable, right? A neuron, if I picked up a neuron at Walmart and looked at it and measured its electrical levels, -70, unstimulated neuron. If I took a skeletal muscle cell and looked at it, it would not be -70, it would instead be negative. What was the resting membrane potential of skeletal muscle? What was it? It's either 80 or 90. I'm going blank right now.
90.
90, yeah, that sounds better, right? So -90, right? So a skeletal muscle has a resting membrane potential of -90. Now what happens? Just think about, you've got a neuron or a muscle, doesn't matter, and if enough sodium comes in, what happens? We reach threshold and boop, an action potential is sent off, right? We get it. Well, it turns out that these cells of the SA node have leaky channels. So they don't have a good resting membrane potential. They instead have a very unstable, their resting membrane potential, if you will, is at -60. So there's another number to keep track of, -60, but is constantly drifting upward because there is slow, leaky sodium gates. So what this is going to do is cause a depolarization, right? So we're thinking about it, the cell's at -60, but because there's leaky sodium channels, what's going to happen? We're going to have an upward curve, aren't we? Right? And we're going to reach a threshold, and that's going to cause an action potential. We're not surprised by that. It's the same old story, except we've never talked about a cell that had an unstable resting membrane potential. So it starts off, where was it again? It starts off at -60. When it gets up to -40, that's its threshold. There's something different here, though. At -40, in addition to the sodium that we've already know and love in this story, there are some voltage-gated calcium channels. We've never had calcium channels showing up in a depolarization system before. So the cardiac, a little different. Got to have calcium around. Part of it is because we need to have calcium for the depolarization of the heart muscle. And this is going to lead it going up to about 0 as a peak. And then as we're accustomed, the potassium channels will then open up and cause the cells to repolarize. So it's really, there's just a couple of different pieces here. The difference here, though, is that the heart is going to naturally go through this depolarization, repolarization cycle every 0.8 seconds. Right? And that's what's setting the heart rate. Every time these cardiac, these SA node cells have their little leaky channels and they reach their threshold and they fire off a signal, that is another, right, another signal sent out from the SA node. Again, every 0.8 seconds or on average 75 times a minute. Let's look at a curve of this. Okay. So you'll notice that there's really never there's no flat line, right? These cells don't have the luxury of having a flat resting membrane potential. So they start off at about -60. There's always a slow sodium leak when they hit -40. Upward swoop, but what's causing the upward? Not only is it an inflow of more sodium, but also of calcium, right? So calcium is rushing in as well. Okay, calcium is, sodium you know is positively charged, 1+. Calcium is 2+. So you've got calcium rushing in, that's creating an even, right, a greater upward, less negative situation. We get to 0, and now some potassium channels now flow out. Same old story. We get back down to the bottom, but because those leaky channels are still there, we immediately start to push back up, and we reach threshold again. So these cells never stop sending their signal. And if you look at the timing on this, right, let's just look, from the beginning of one to the next one, right? We finish the cycle in what? About 0.8 seconds, right? So every 0.8 seconds, this whole cycle of depolarizing is going to occur. This is how those pacemaker cells do their job. Does that graph make sense? Okay, the only real difference here is what? A couple different numbers and the presence of calcium. So, right, that means that the SA node is constantly depolarizing, and the two atria are going to simultaneously contract, almost simultaneously, right? Essentially, they're going to contract simultaneously. And it's going to take about 50 milliseconds for the signal to go from the SA node over to the AV node. 50 milliseconds. What's that look like to you? Fast, right? Really fast, right? So when you think about so I'm going to draw my beautiful drawings that I do. So there's the SA node, and we saw that signal go to all places, didn't we? Spreading all around the right atrium, and it goes down to the AV node. How long is this going to take? 50 milliseconds. That's all it's going to take, very, very quickly for that signal to get down there. The problem is, though, the cells in the AV node have fewer gap junctions. They have basically a less developed intercalated disc. So even though the signal gets to them very, very quickly, it takes a little bit longer for the ventricles to get that signal. There's a delay. So even though the signal got there in 50 milliseconds, there's another 100 millisecond delay. Well, that's all good. Because what's going on? I need us to think about the heart for a second. When the atria contract, right? Where's that blood going? Down through the AV valves to the ventricles. So when the SA node fired, what started? The atria started to contract. We better give the atria time to squeeze, and we better give the ventricles time to fill and receive that blood. Because if we too quickly start squeezing the ventricles, right, there wouldn't have been time for the blood to go from the atria down to the ventricles. So the heart has this little hiccup, or this little delay in it, if you will, between the atria and the ventricles, and it's necessary. If all four chambers were squeezing at the same time, or they weren't well coordinated, then the heart and the blood would not travel properly. Now in lab today, and this week I'll introduce that when all of the cells within the atria contract basically at the same time, we call that a syncytium. And if you break that word down, there are two Y's in it. It's kind of a funky word to spell. But if you break that word down, syn meaning together, and site. So all the cells are working as a single unit. So all of the cardiocytes within the atria are going to squeeze essentially together, and then all of the cardiocytes in the ventricles in a moment will also squeeze as a functional group, as a syncytium. So once the signal gets from the AV to the AV node, right? I told you there was a delay. It takes a while for the AV node to sort of get activated, but then once it gets picked up by the AV node, then it's going to travel very quickly down the bundle branches, down the Purkinje fibers, and as this signal is traveling down, now the ventricle will be in a depolarizing contractile phase. Again, nearly all the ventricular cells will be contracting simultaneously, the idea of a syncytium. Also, remember that the Purkinje fibers go up and interact with the papillary muscles, which are going to basically pull, right? They're going to contract, and as those papillary muscles contract, what's attached to the papillary muscles? The chordae tendineae, right? And so as the papillary muscles contract, they're going to pull on those chordae tendineae, and what are those going to do? Keep the valve. What valves? Which valves are attached to the chordae tendineae? the AV valves, right? The right and left atrioventricular valves, the bicuspid and the tricuspid. And so they're going to keep those valves nice and tight so that blood can't go back up into the atria. Also, as the ventricles are squeezing, as they're in systole, it's going to go from the apex up toward the top of the heart, and it's going to do so in that twisting motion. So again, those cardiocytes are in sort of that twisted arrangement. So when the ventricles squeeze, it's going to twist like you're wringing out a mop and push that blood up and out the semilunar valves. Now that graph I showed you a moment ago is only the graph for the depolarization of the SA node cells. Okay? So what were the numbers there again? Negative 60 and negative 40. Okay? That was just those cells called the SA node pacemaker cells. But the rest of the cardiac muscle is also excitable and also has to undergo its own depolarization event. So here I'm going to talk about the little bit of difference here, and then we'll take a quick break. So regular heart cells, right, regular cardiocytes do have a stable resting membrane potential. Their stable resting membrane potential is negative 90, same as skeletal muscle, right? Same starting place as skeletal muscle. So that means that the rest of the heart will only depolarize when it is directly stimulated. There are no leaky sodium channels in most of your heart muscle. So the signal comes from where? The signal is going to come from the SA node. That depolarizing event is now going to do what we already know and love. It's going to cause a depolarization of the muscle cells. They will peak up to positive 30, and we know what happens, but there's a difference here, and this is a really, really cool difference. If I'm going to draw a normal action potential, right, resting membrane potential, up toward threshold, boom, up, depolarizing, back down, a little hyperpolarization, and back to normal, right? You agree? In the heart, the cardiac cells do this. Resting membrane potential, move toward threshold, go up, but then they plateau, and they stay up there for a long time. That plateau is going to be between 200 and 250 milliseconds. What's happening during that? Okay, what's happening during that plateau? This is when the heart is maintaining a sustained contraction. If this is skeletal muscle here, what do we call a rapid depolarization, repolarization? What's the word? Starts with T. Twitch, right? Twitch. So a twitch, right? Quick movement of skeletal muscle. Do we want your heart twitching? We don't want your heart twitching. We want your heart to, when it squeezes, to keep on squeezing, get all the blood out, and then relax. Squeeze, sustain, and relax. So as a result, there's a plateau. So this plateau is, you don't want your heart cells going up and down, twitch really quick. You want that long, sustained contraction. And this plateau is exactly that. This 200 to 250 millisecond plateau is the time during which the cells are in a sustained contraction. Now, what keeps it up there? Remember those, I mentioned some calcium channels? They're still around. There are calcium channels that are very slow to close, and the sarcoplasmic reticulum is not very fast at sequestering the calcium back. So now we have to go back. Remember I told you that the action, the whole actin, myosin story of skeletal muscle is very, very similar, not exactly, but very, very similar to cardiac muscle. So we still have T-tubules, we still have sarcoplasmic reticula, we still have all of that story going on in the cardiac muscle. So we've got calcium channels that are slow to close. That means that what? It's going to keep the value up high, isn't it? And the calcium doesn't get quickly sequestered which is going to, again, keep that muscle contracting longer. It doesn't get sequestered out like it did before. So eventually, after that long plateau, then there will be a repolarization. And the repolarization will be when the calcium channels finally close. And of course, those potassium channels that we know and love will then open and return the cell back to its negative 90 resting membrane potential. Therefore, there is in cardiac muscle a long, absolute refractory period of 250 milliseconds. Well, that's only a quarter of a second, right? But that's a really important quarter of a second. Skeletal muscle only has a refractory period of 1 to 2 milliseconds. Remember when we were talking about how the stair-step effect? Right, you could have rapid stimulation of a skeletal muscle to the point where you could have multiple really, really quick stimulations, and the skeletal muscle wouldn't have the chance to come back to resting. But you recognize that those twitches can be very, very fast. Depolarize, repolarize in just a couple milliseconds. Heart muscle, none of that. You cannot get the heart to refire again during that absolute refractory period. So that word means exactly like it did in the nervous system. The absolute refractory period is the time, no matter what you do, the heart cannot enter into another action potential. So there's going to be a break in time. Well, what this does is it prevents summation from happening in the heart and prevents the heart from ever reaching tetanus. Does that make sense? So what can happen to skeletal muscle? Stimulate, 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 and what happens? You start getting a stair step, and eventually you get up to tetanus, right, where the cell is in total contraction and it can't relax. Because of this absolute refractory period, the heart muscle, the cardiac muscle, will never go into tetanus. It will never allow for this constant summation because of this relatively long absolute refractory period. So look at these. In fact, I'm going to have you take a break now, and we'll come back and we'll look at these two curves. One is actually the action potential, and one is the actual contraction of the muscle. But let's take a couple minutes, refresh the minds and the body, and we'll come back and look at this chart together. Okay, any questions so far? Anything at all that comes to mind? I'm expecting great things on this test.
Yes.
I think the skeletal muscle story makes sense to you. I think you've had time to let that sink in. I've heard someone say that the quiz is a little bit long on skeletal muscle. I'm seeing a few affirmations, so a little long, but with more questions comes more practice, comes better understanding, comes higher test score. Right. So that's the plan. And as you're bumbling, whatever the word is, through those test questions and those quiz questions, you are learning, right? So it's a great way of preparing. So definitely do your Connect assignments followed by the quiz. Remember, it's open book, open resource, so you can take your time on it. Then there'll be a blood quiz that's also open now. Listen to the YouTube video on blood. Do the blood Connect questions and do the quiz. I think you'll find that to be very straightforward. And then there'll be the third quiz on the heart. Those quizzes are all due to turn off at Sunday midnight. So no wailing and gnashing of teeth and emails, I didn't know, right? Sunday at midnight, because it is a Tuesday exam. That'll give you Monday to review it. Remember that if you don't start the quiz, you don't get to review the quiz. So if you don't even start the quiz, you won't get to look at the correct answers. If you did at least start it, then you will get to look at the correct answers. Okay, so let's take a look at this double curve, if you will. I know it's small, but what you're seeing on here is the one curve. The green is the action potential. So again, we are at negative 90. Reach threshold. And then there's this long plateau. And that plateau, again, was how long? About 0.2 to 0.25 seconds, right? So that makes sense from what we're seeing here. Right, there we go. About 0.2 seconds. But what's going on with the muscle? So that's the action potential, but as you can imagine, there's going to be a slight delay from the onset of the muscle contraction, and then it's going to come up and down. But again, notice that this is a rather wide time of muscle contraction. So there's a very long, sustained contraction that goes on with this. What's driving this, again, is sodium going straight up. And then there's going to be some slow calcium channels that are going to slowly close. It's going to keep it up high. And then eventually the potassium channels will open and bring this back down in a repolarizing way. Again, your resting membrane potential is very, very stable for a normal cardiac myocyte. So do we appreciate the two different curves? The first curve I showed you was for a pacemaker cell, right? The cells that never have a resting membrane potential, have those leaky channels, and are constantly re-depolarizing, right? Is that the right word? Constantly depolarizing again through those leaky sodium channels. And then the rest of the heart is made up of cells that have a very stable resting membrane potential and only depolarize when activated through this signal from the SA node. So when all this electrical activity is zipping around in the brain, in the heart, we can detect that using an EKG or an ECG. So I was trained with a person who used EKG, so I tend to say that more often naturally, but I'll flip back and forth. They're exactly the same thing. The K from the German for cardio with a K. So English or German, doesn't matter. And what you'll see in lab and what I'll introduce to you again today is that there's a normal sinus rhythm. That within a normal EKG, there's going to be a P wave. There's going to be what's called the QRS complex of rather significant disturbance. And then there's going to be another wave, the T wave. Then there'll be a gap of time before the whole thing starts all over again. And in lab, we'll recognize that from one curve to the next, on average, 0.8 seconds. So when I think about R to R, this R wave to R wave, 0.8 seconds represents what? The average time for the cardiac cycle. And if one has their heart beating every 0.8 seconds, what is their heart rate? Those in lab have already been through this today, but if you have a heart rate, or if you have a beating heart every 0.8 seconds, what is your heart rate? Let's think about this another way. If your heart rate was every 1 second, then your heart rate, or if your heart beat every second, then you would have a heart rate of 60. If you have a heart rate, or if you have a beat every 0.8 seconds, then your beat per minute would be what? The answer is 75. And how do we get that? Not times. 60 divided by 0.8. And that will give you 75. Okay, and in lab we'll talk about a couple other ways of measuring your heart rate using an EKG. So let me clear this and we'll go through this. The P wave, and it tells us down below, the P wave represents this upward movement, this upward disturbance represents the depolarizing of the atria. So the atria are contracting. And they're going to be contracting all the way until about here. So you see this bar. So the atria are contracting here. And then the QRS complex represents a much more substantial electrical disturbance because this is where the ventricles begin to contract, and they actually contract not just during this little period of time, but they're contracting during this period of time, right? So they're contracting during this box. Sustained contraction. And then the T wave represents the repolarization, the relaxation of the ventricles. So let me walk you through this again, slide by slide. So you'll recognize this from lab. The P wave represents the electrical disturbance caused by the firing or the depolarization of the SA node. The atria, in this point, those cells are depolarizing. As those cells depolarize, they are contracting. We would call this atrial systole, and there's going to be a slight delay, right? About 100 milliseconds. So from the time the SA node fires until the atria are contracting, 100 millisecond delay. You might better write that as 0.1 second. A tenth of a second. Then there's time. I told you that there's about 50 milliseconds. Now, I've given you this information in different places. Remember I told you there's about 50 milliseconds for the signal to travel from the SA node to the AV node. And this will set up now the ventricular depolarization. And you'll see that huge spike. And the spike, the size of the spike has to do with the massive thickness of the ventricle. It's just a much thicker, much more strong contraction. There's a time called the S to T segment. This is the time from the end of the ventricular depolarization until the ventricles begin to repolarize, and this is the entire time during which the ventricles are contracting when they're in systole. This is also the time on the action potential where you would have the plateau. And then finally the T wave will be formed. That is caused by the electrical disturbance of the ventricles now repolarizing and therefore relaxing. So let's take a look at this again. P wave. Again, that electrical disturbance represents what? The SA node is firing, isn't it? And right after the SA node fires, what will happen? The atria will begin to contract. We would say the atria are in systole. They're contracting. During this entire time, what's happening where I'm drawing the bar? That's the time during which the atria are contracting, right? They're a sustained contraction. Then the electrical signal will have gone from the SA node down to the AV node. The initiation of the AV node will now cause the ventricles to begin to depolarize. That electrical disturbance is shown here. Again, that's the AV node now. And now the ventricles will be contracting, won't they? And they'll be contracting during this boxed time. So the ventricles are now in systole. And then there'll be a disturbance of the T wave when there's a repolarization of the ventricles. The one event that we cannot really see very well on an EKG is the atrial repolarization, right? We don't see a separate wave for the atrial repolarization. That disturbance is actually hidden and is overwhelmed by the size of the ventricular contraction. So you don't see it, but be assured that there is some electrical disturbance from the atria repolarizing. So we'll break this down a little bit. This figure does come from your book if you want to look at it more carefully, 1916. But if you look at it, again, this just shows you the waves compared to what's happening in the heart and walks you through it. So again, the P wave, what we see here in number 1, the P wave is because the atria are undergoing depolarization. They will continue to contract all the way until the QRS begins. Oops, going down here, sorry. They'll continue to contract through until the QRS is beginning. And at that point now, the ventricles will begin to depolarize. The atria will be relaxing. The ventricles will then continue their depolarizing, their contractile phase. They'll eventually begin to repolarize when the T wave is showing up and return back to a relaxed state. And now we're back to that baseline. So I'm going to point to a portion of the wave, and I want you to tell me what's going on. So P wave. Popcorn out, what's going on? P wave. What's firing? SA node. As soon as the SA node fires, the atrial cells will begin to depolarize. And and the cells, even though the P wave is quick and goes up and comes back down, the atria will continue to be in a contractile phase until when? Pretty much the beginning of the QRS. At the beginning of the QRS, what is now firing?
Ventricle.
The AV node is firing. The ventricles will begin to contract or go into systole or go into depolarizing event. What we don't see is a wave for the relaxation or the repolarizing of the atria, but it's under there. So then what does this time represent? That pretty much represents the time during which the ventricles are now in a sustained contraction, again because of that plateau of the action potential. And then the T wave represents the repolarization and therefore the relaxation of the ventricles. And what does this time in between here represent? A time of complete relaxation, a time of diastole. And what's happening during this period? What's happening at the SA node? Those leaky sodium channels are leaking, aren't they? And they're slowly going to cause another what? Signal to be sent, which would initiate another P wave of the next wave, of the next cardiac cycle. So you'll look at a number of normal and abnormal EKGs in the lab, and you'll recognize that this is a normal sinus rhythm. Now you can't, there could be some things wrong with this, but when you look at this wave, you can see, I think even though it's small, you can see the P wave, you can see the QRS, and then you can see a T wave at the end of each. So it looks rather normal. And you'll learn in lab how to measure the distance and the time that's going on during this. Now take a look at the lower one. What curve is, or what peak is missing? Yeah, do you see there's no P wave? So we do see a QRS, and I do see a T, but there's no P wave. So therefore, what's not happening? There's no firing of the SA node. Okay. Now I told you if there's no firing of the SA node, that the heart would beat closer to what? 40 to 50 times per minute, and pretty confident that if you looked at the time between these R to R waves, you would find that the heart is definitely beating more slowly than in a normal heart. Now, what would cause an abnormal EKG? All sorts of things, right? It's a very quick screening tool that we can use in the clinic. Have all the leads put on, have an EKG done. In just 6 or 10 seconds of data, you can tell a lot about one's heart. So it takes longer to set the EKG up than it does to actually perform the test. If you've had an EKG done, you know, they sit there and they put the 12 leads on you, and there's all that production of putting everything on, and they push start, and before you know it, they're ripping the electrodes off you, and they have a short time of data, which is then read partially by the computer, mostly by hand. And if there's something significantly wrong, right, something's off, then they'll come back and do a more complete study. But a normal EKG is just a screening mechanism. But what sorts of things can an EKG notice or measure? It can see problems in your conduction pathway. You can see if there's a problem in your SA node firing, a problem in your AV node, a problem in the conduction between those sites. It can tell if you've had a heart attack, because there'll be some differences in the strength of the heart and the way it behaves. You can tell if the heart is enlarged. You do not want an enlarged heart. It's the one muscle in your body that you want to keep small. You want a nice, tight, efficient heart. heart enlargement is a sign of a less efficient heart. So as the heart enlarges, and it does enlarge a little bit with age, but you want a nice, tight, efficient, highly, well, efficient's the best word for it. So when it pumps, it's doing so with great force and with great efficiency. And a large heart cannot work as well. And also, if you've got some significant hormonal or electrolyte imbalances, that also would show up because those electrolytes are going to influence the ability of the heart to depolarize. So you'll see some hiccups, if you will, in the overall conduction system if you have some hormonal or electrolyte imbalances. So you do a quick EKG, right, and we can tell, and then do some further studies if something seems to be abnormal. Stop there. Any questions about EKGs? Those who were in lab today, do you agree that that was pretty straightforward after what you saw in lab? And those who have lab tonight or Thursday, you'll get the whole story. You'll feel much more comfortable with that little section before we have our test next Tuesday. Stop there. Any questions at all? Okay. Well, continuing on with the chambers of the heart, we recognize that each cycle, each beat of the heart is a cardiac cycle, which is a complete contraction and relaxation cycle. It's going to involve the contraction and relaxation of all four chambers, not at the same time, of course. There will be first the two atria that will contract, the atrial systole, and then shortly thereafter there'll be the two ventricles that will also contract. There is going to be a time where there's a quiescent period. So where did we see the quiescent period? Going back, where nothing's really going on. Where is the best period where nothing's contracting or really relaxing? Everything's kind of at rest?
Between the P and the Q.
Do you agree between T and P? Right, during this period of time, that's when really nothing's going on. All four chambers are relaxed. And so we would call that the T to P interval. So we're going to be going into a little conversation about blood pressure. And I've already told you that the chambers of the heart do not open unless they're forced open through a pressure change. We have to go back to a little bit of basic physics. That may scare you. Nothing really heavy duty here. But you all experience, if you have water in a hose, does it flow unless there's something pushing it? Right? So nothing's different there than in your body. So if you have a blood vessel and you want blood to be pushed through that blood vessel, there better be something pushing it. There better be a pressure gradient. Right? So we'll use the word pressure gradient. You'll hear the word fluid dynamics. You can take entire courses if you're going into engineering or some sort of field where they talk a lot of physics. You would talk about fluid dynamics. But basically, for a fluid to flow through a tube, there has to be pressure pushing it through that tube. And by the very nature, there will be resistance that will push back and fight the movement of the fluid. So the pressure gradient is simply the pressure difference between the front and the back end, right? Point A and point B. This is going to be measured in millimeters of mercury. How convenient. Those are the same units used to measure blood pressure. All right, so we're going to measure this pressure difference in vessels and in the heart with millimeters of mercury. And we're going to use a sphygmomanometer, which is a blood pressure cuff, okay, to measure blood pressure in the lab. When fluid is being pushed or blood is being pushed through the vessel, there will be a natural amount of resistance that is also opposing the fluid flow. Does it make sense to you that the bigger the vessel, the bigger the diameter of the vessel, the less resistance? And if I have a very narrow vessel, that narrow would increase the resistance, but what else would it do? It also would, if I'm going to squeeze on a garden hose, right, as I squeeze on the garden hose, what happens to the pressure?
Expands.
Water going out is going to be under greater pressure, right? Because I'm going to squeeze. So we'll see this relationship between pressure and resistance. And again, I'll say it in words. Fluid will flow down its pressure gradient. It'll always go from an area of higher concentration, or higher pressure, sorry, to lower pressure. That's just the pressure gradient. You want water going through your garden hose, you better have water pumping out, right, pushing that water through the hose. So this pressure is what is going to be pushing your valves open. So let's think about the left side of the heart for a second. Left side of the heart, bigger, stronger side, right? And when the ventricles relax, they're going to expand. Just imagine the ventricles are, they were contracting, now they are relaxing. And as they relax, they're going to expand, and therefore the volume of the ventricles would do what? Get larger, and the pressure would fall. There'd be no pressure. Now, if the bicuspid valve is open, which one's the bicuspid? That's also called the left AV valve, right? What's going to happen? If the left AV or the bicuspid valve is open, then blood is going to flow from where? From the left atrium down into the left ventricle. No problem. Then the ventricle is going to contract. As it gets smaller, the pressure is going to do what? Increase.
Yeah.
And eventually, right, what will happen? The AV valve will close. Why will the AV valve close?
The blood doesn't go back.
We don't want blood going back, right? We don't want blood going back up into the atria. And the aortic valve would be pushed open. Why? Because of the increased pressure. And push that blood up and out of the aortic valve, leaving the heart. So the AV valves basically go limp when the ventricles are relaxed, and the semilunar valves are under pressure when the ventricles are relaxed. Okay? That makes sense? So when the ventricles are squeezing, which valves are are open, I should say. Which valves are open? Yeah, the semilunar valves, right? And they're under pressure, and so the blood is being pushed out. So let's just take a look at this. Let's make sense of this. So blood's coming in to what? Superior and inferior vena cava. We're in the right atrium. Blood then goes down into the right ventricle. It got there through the tricuspid or the right atrioventricular valve, right? It's kind of a quick review of all this. Simultaneously, what's going on? Blood from the left atrium is coming down through the left AV or the bicuspid or the mitral valve. So the AV valves are open, blood is coming down. And then when the ventricles begin to squeeze, then what's going to close? Those AV valves are going to close, and now what's going to be pushed open will be these semilunar valves pushing blood up into those chambers. And then what happens? It can't come back, can it? So we think about blood being unidirectional. There's a little bit of, back and forth going on as it pumps, but for the most part, blood is moving unidirectionally, as we just described. Now, what if the valves are messed up? There's some sort of valvular insufficiency or some sort of incompetence. Nowadays, we can put in some artificial valves, we can put in some pig valves. We can do amazing things now with the heart when things don't go right. But if the heart is no longer, if the valves are no longer holding their job, then blood can go backwards. If blood is going backwards, then the heart is not as efficient as it should be, and not enough blood is getting to its proper place. Under the stethoscope, if you listen to someone who has a backflow, we call this a murmur. You'll hear a regurgitation of the blood. It's almost like a little gurgling, okay? And the ventricles are squeezing, but blood is going back up. Now, the valves that we're mostly talking about here are going to be the AV valves, because those are the valves that are under the greatest pressure. As the ventricles are squeezing, those are the valves that have to keep things closed so things don't go back up into the atria. So that's usually the place you're going to see a valvular insufficiency would be where? Which valve is under the greatest pressure? Which one's under the greatest pressure? The bicuspid or the mitral or the left AV? Why? The left ventricle is the strongest chamber, and so as it squeezes, right, and pushes blood out the aorta, that left AV valve is also under greater pressure. So that's the one that's more likely going to become leaky if there's going to be a problem. Now, what can cause this? A lot of things. There can be scar tissue that can happen, and that scar tissue would not allow the valves to close properly and tightly. Rheumatic fever is a common issue here. Rheumatic fever, a strep infection, which can lead later on in life to an autoimmune attack of the valve cells. The heart can be overworked and become enlarged, and no longer do the valves work as they should. And again, whenever you have this backflow, this incompetent valve, this regurgitation, we refer to this as a murmur. Most common one, again, mitral valve, and you'll hear the word prolapse. Mitral valve prolapse. So basically, when the ventricles are contracting, blood is going back up into the left atria. This is a relatively common situation. There's hereditary forms of this, so it kind of runs in families. The good news is we can do a lot to fix it if it becomes a problem. And if you didn't have sufficient blood, what would you expect the symptoms to be? If you weren't getting enough blood forward, right, and you had too much blood going backwards, then you would not have sufficient oxygenation. So you would complain of shortness of breath and tiredness and not being able to and maybe even chest pain. And at that point, definitely we're going to get you in and fix your valves, right? So they may be watching a murmur. You may have been told, oh, you have a slight murmur, we'll just watch it. And every year you go back and they say, oh, you have a slight murmur, no problems. But at some point in the future, it might become a problem, in which case they would take surgical procedures as an option. What sounds does the heart make? Lub-dub, right? Lub-dub, lub-dub, lub-dub. And those sounds, lub-dub, lub-dub. Those sounds are caused by what? Yeah, the lub-dub, the sound you're actually hearing is actually the percussive closing of the valves, right? So lub is closing of what? AV valves, and dub, the semilunar valves, right? So we're going to just change these from lub and dub to S1 and S2, and their lub and dub is fine. So S1 is a louder, longer sound. It is from the closure, as you already guessed, of the AV valves. It's also, in addition to just the percussive closing, it's also the blood, right? Sort of bouncing off. So the blood was coming down and all of a sudden, oop, it was stopped. So it's blood kind of running into the valve, running into the chamber, and that turbulence of the bloodstream causes this collective thing called lub or S1. The second one, dub, is a softer, sharper sound. Again, it's the closure of the semilunar valves along with the overall movement and the sudden stopping of the blood. There is an S3. I'm not going to worry about it. It's not usually an issue in people over 30 and not something I'm going to worry about. Now, you'll see arguments in different textbooks if you were to look about what actually causes these sounds. Is it truly the percussive closing, or is it some other artifact or some other phenomenon? Okay, just know there's a controversy at the real world of what this is, but we'll use the combination of all of these things to give us the sound, right? It has to be a combination of the closing of the valves, the flow of blood being stopped suddenly, and the overall movement of the heart wall. So we know we have heart sounds, S1 and S2, or lub and dub. And I'll take the last few minutes and we'll go through the phases of the cardiac cycle, and then I'll pick up again with reviewing this on Thursday when we meet again. So this is what's going on. Now, to picture this, you definitely need to know your chambers. You definitely need to know your valves, and that cannot be something that's confusing to you. So again, I'm gonna beg you, if that's something you don't know really, really well, before you study any of this or before you take the quiz or do any of this, you really do need to go back and review the overall anatomy of the heart. So phases of the cardiac cycle. We're going to start off with the filling of the ventricles. This isn't hard. The ventricular filling. Well, what's happening during, I'm going to give you more details, but what's happening during ventricular filling? They're filling because what's squeezing? The atria are squeezing, they're in systole, and what valve is open? Valves. The AV valves, right, are open, allowing for the ventricles to fill. We got it. And then the ventricles will begin to contract in what's called isovolumetric. Iso, same, volumetric, same volume. And during isovolumetric contraction, the ventricles are beginning to squeeze, but the volume hasn't changed yet. It's still the same volume. Hold on. Then, at some point, you're going to squeeze the ventricles strong enough that you're going to push open what valve? Semilunar valves. And now there's going to be an ejection, right, of the blood in the ventricles. And then the ventricles will begin to relax in an isovolumetric way again. So in other words, they're going to relax, but there's no new blood coming in, the same amount of blood, isovolume. And then all over again, what's going to happen next? Then the ventricles are going to start doing what? Filling again.
Yeah.
Now this whole thing, we'll break it down, is happening again every 0.8 seconds on average, less than 1 second. So like so many things we've done this semester, from action potentials to muscle contractions, we slow this down like it's in molasses, but these are all very rapid processes that we're learning the steps about. So let's go through these. We'll see a picture of it, and that'll bring us to the end of our evening. So ventricular filling. We've already said this, but now it's in words. Let's check our knowledge. So during this time, now I'm saying that it's during diastole. Don't be confused. What's in diastole? During ventricular filling, this is not atrial diastole, this is ventricular diastole, right? When the ventricles are relaxed. So they're sitting there just open, waiting. And during this time, the ventricles are expanding, the pressure is dropping, the AV valves are open, and the blood is flowing down into the ventricles. Okay, all right, so we're picturing it. Spigots open, blood's falling, largely by squeezing of the atria and gravity. The atria are not very muscular. When you do your heart dissections next week, I think you'll be reminded of how weak the atria are compared to the ventricles. So yes, they're squeezing, but it's largely also just dropping down by gravity. In fact, we're going to break this into 3 phases. There's rapid ventricular filling. Pretty much gravity, right? So as the valve opens, poof, drops down. Then there's going to be the second 1/3 of time, and this is a slower filling of the ventricles, and this is called diastasis, and this is, the P wave occurs at the end of this diastasis. P wave. What's the P wave? The SA node, right? And so we'll have to, in a moment, what you're going to have to do, really what you need to be able to do, and I'll show you a figure of this, is to put all these different events into your mind in an overlapping way. We've talked about the conduction system. We've talked about what that looks like on an EKG. Now we're discussing, and we talked about the action potentials that are happening from the SA to the AV node. And now we're talking about what's physically happening in the heart during these different times. So you kind of got to be able to kind of superimpose these 3 or 4 different stories into one cohesive page or one cohesive story. And then finally, the atria are going to squeeze. So what happened? The valve opened, blood quickly went in, and then it started squeezing, and finally it squeezed out. Basically, I always think about it, it finally squeezed out the last little bit. Atria contract during atrial system. Now, at the end of this time, we've just filled the ventricles, right? So you're picturing it, the ventricles are sitting there, right? They got all the blood in them they're going to have for that cycle. The amount of blood that's in the ventricle at that moment is called the EDV, the end diastolic volume. It's just the amount of blood sitting in the ventricles before they squeeze. On average, 130 mils. So there's going to be 130 milliliters of blood sitting in your ventricles before they begin to squeeze. At the end of diastole, at the end of the relaxation, they've just been filled 130 mils of blood. The ventricles are then going to begin to contract. They're going to contract. Why? I mean, the atria have repolarized, they're relaxed. And the ventricles are going to depolarize, and they're going to begin to create the QRS complex, and they're going to begin to contract. But there's sort of a slow ramping up to the contraction. So for a while, although the heart is in a contractile phase, the ventricles are, there's no change in volume. So it's called the isovolumetric contraction. The AV valves are going to close, and when those AV valves close, when those AV valves close, you're going to hear the S1. Again, isovolumetric because even though the ventricles are contracting, they haven't yet spit out any blood, and the volume is still the same. At this point, this should be cardio, not cardio. The cardiocytes are exerting force, they're squeezing, but all 4 valves are closed for this brief moment, and there's no place for blood to go. At some point, the pressure in the ventricles is going to reach a point where it's going to force those semilunar valves open. Blood is going to start being spurted out of the ventricles rapidly at first and then slower. So there'll first be a rapid ejection and then a slower reduced ejection. This ejection is going to last for 200 to 250 milliseconds. Okay, that 200 to 250 milliseconds corresponds to the plateau phase we saw in the cardiac action potential, right? That long, sustained squeeze is going to be happening. So the ejection of blood will happen for that sustained squeeze. We'll then continue to eject blood from the ventricles. The T wave will occur. That means that the ventricles are going to start to repolarize or relax. How much blood the heart just ejected is going to be called the stroke volume. So I just squeeze, right, the ventricles, and on average, I'm going to squeeze 70 milliliters of blood out. How much was there in the beginning of the contraction? 130. Only 70 gets squeezed out. And we're going to call this the ejection fraction, the EF. On normal heart, about 54%. Where'd that number come from? 70 out of 130, right? About 54% of the blood, on average, is going to be ejected when you are at rest. When you're in exercise or the bear is chasing you, not only is your heart beating faster, but it's beating stronger. And so you would be potentially spitting out 90% of the blood at any one stroke. So the stroke volume, the SV, is the amount of blood that you are stroking out, right, with each stroke, with each contraction of the heart. Next lab, we'll be looking at this equation and doing some activities with stroke volume. Lastly, after you have pushed the blood out, what's left in the ventricle is now called the ESV, the end systolic volume, the volume at the end of the contraction. And this is going to be 60 mils. So these numbers all make sense, right? You start with 130, you squeezed out 70, that's the stroke volume, and you left 60 behind. That's called the ESV. So we'll pick up with this. I'll come back and walk you through this phases again. We're on slide 90. There are only, I say only, but that leaves us with 40 slides left, right, to 130 to get through this. So I'm very, very confident that we'll get through the rest of this material very comfortably. I also would beg you, if you have any concerns at all, that you come on Thursday with some formulated questions related to skeletal muscle, to blood, or to cardiac, so that I can address those with you on Thursday.